let's talk about adaptive immunity. So, on a good day, your innate immunity, the one that you're born with, is going to be able to deal with minor infection. Right? You're going to get better very quickly. There is no need to involve adaptive immunity. However, in cases where you come down with something that's really difficult to deal with, like again, chickenpox, hepatitis, flu, cold, right? All these things, the innate immunity is not very good with dealing with it, and that's when you need to bring out the big guns, the adaptive immunity. Okay, so adaptive immunity is going to create long-lasting mechanisms that are going to be tailored to target a specific invading pathogen. And typically speaking, once you have created the adaptive immunity for that pathogen, you are going to be protected against it for life. And we're going to look at how that is possible. First, let's talk about some characteristics of adaptive immunity. Right? Number one is there needs to be a way to distinguish self and non-self. Right? You want to know what to attack. Right? You don't want to indiscriminately attack everything. Right? You want to know who's the enemy. So this is a characteristics of adaptive immunity. Another one is memory. Okay, you have the ability to remember a specific foreign antigen, right, on the pathogen, and so next time if you encounter it again, you are going to be able to deal with it much quicker. At this point, maybe I should clarify. The difference between pathogen and antigen. Right? Some people might find that confusing. So pathogens are basically living things that cause you to become sick, right? So bacteria, right? Bacteria is a pathogen. Virus, although virus, some people don't consider them as living, but it's an example of pathogens. Fungus, right? Parasites, so all these things are pathogens. They can cause you to become sick, right? And they are for the most part alive, right? And antigens are not necessarily bad, right? Antigens are just molecules that are found on the surface of a cell. So you remember your cells have antigens, right? In fact, your red cells have antigens that determines your blood type. So it's not a bad thing. It's just something that we all have, right? All cells have it. So bacteria, virus, and fungus, they also have antigens. And so what we're talking about here is if your immune system sees a foreign antigen, something that your body does not have, then it's going to react against it, right? You're going to create immunity, you're going to create antibodies to attack that foreign antigen. So let's say we have a bacteria here, and there is an antigen X here on the bacteria, okay? And we don't have antigen X in the body, so when your immune cell sees this one, they are going to create an antibody to attack this foreign antigen, which is found on the bacteria, right? And so next time when you come down with the same thing, you will remember that you fought off this foreign antigen before, and so you're going to be able to respond to it much quicker. Another characteristics of adaptive immunity is specificity and also diversity. So specificity, I'm just going to read this to you first, is the ability to respond and to combat specific antigens, whereas diversity means potentially you can respond to billions of different antigen receptors. So it's a little bit confusing here, but you can think of having a whole set of screwdrivers. Okay, so the screwdrivers, the tips are different for each screwdrivers, but then the handles are pretty much the same thing. So each individual screwdriver is designed to tighten a particular type of screws. Okay, so like if you have a flat screws or something, then you would need the one with the flat head. Right? If you have one with a cross or something, I don't know what they're actually called, then you would need the one with the cross. So each screwdriver is only good for one type of screws. Okay, but if you have the whole set, then you can basically tighten any screws in existence, right? So that's kind of like that. Individual antibodies are very specific. Okay, each time your adaptive immunity is going to create a tailored response for one type of pathogens. Okay, and that will only work for that specific infection. But collectively, your adaptive immunity can create many billions and billions of different types of response that would be useful for potentially billions and billions of different types of pathogens. So central to adaptive immunity is the ability to generate antibodies. Antibodies is like the final ultimate weapon we have to defend the body. If the antibodies does not work, then you're not going to get better. Okay, and that's when you need to have the medication to help you, right? So the basic unit of an antibody is basically a Y-shaped proteins, and I mean we learned about this when we discussed blood transfusion, right? So this Y-shaped protein has two parts. Okay, the trunk of the antibodies we call that the constant region. Okay, they are pretty much the same for all antibodies. You can think of that as like the handle of a screwdriver. And then you have the arms. The arms is what we call the variable regions. Okay, and that would be like the tip of the screwdriver. And each of the antibody will have a different variable regions designed to bind to a specific antigen. There is a key and lock relationship between the variable regions and the antigen that it's designed to bind to. Okay, so over here we have little pentagon groove, and that would only fit for the pentagon antigens here. And here we have little semicircles, knobs, and that would only work for the semicircle antigens for the bacteria. So each antibody is specific to its a target. So that's the specificity characteristics that we were talking about. However, you can make many different types of antibodies. You can make billions, potentially billions of different types of antibodies for all the different types of infection that you are going to experience in your life. And that ability to create all these different types of antibodies is the characteristics we call diversity. Okay, so that's what antibodies are. Let's take a look at some of the ways that antibodies work to help with clearing the infection. Antibody reacts with viruses and toxins. Toxins are basically poisons made by bacteria. Sometimes it's not the bacteria themselves that cause you to become sick, but it's the toxins that they produce that makes you sick. So, for example, in botulism, you can cook your food 100%, but the heat will not destroy the toxin, and if you consume the food, you will still get food poisoning, right? So antibody works by coating these viruses or toxins, and it will cause them to become neutralized. So this is called neutralization. So here you can see some toxic molecules produced by the bacteria, and if you expose your cells to it, then it's going to damage the cell and potentially destroy them, okay, making you sick. But in the presence of antibodies, the antibodies will bind to the toxin and neutralize them, so they are not going to be able to attach to your cells. So that's a good thing. Here we have some red cells, and we have some antibodies and some virus, and the antibodies will attach to the virus antigens, and they will no longer be able to attach to your red cells, right? So they neutralize virus, they neutralize toxin. Here's another one. Antibodies can bind to the target and form an immune complex. We know this already from our discussion on cross reaction. Remember, if you have a cell with antigen A and you mix it with an anti-A antibody, what happens is the antibody will bind to the cell and causes clumping, right? Agglutination. So this is the exact same concept, except instead of having a red cell, you're going to have a bacteria, and let's say the bacteria has antigen X on it, then your antibody will be anti-X, and it will bind to the target and cause them to clump together. So this big huge clump that you get after the antibody binds to its target, that is what we call an immune complex. Okay, so these big huge immune complexes will be engulfed by phagocytes. Okay, so here you have macrophage, and it's eating up all these immune complex, right? So it's basically just put them all together in a big chunk, so it's easier to see, and you can eat more at the same time. Next one, cells can be coated with antibodies, and that will actually activate complements and encourage them to form membrane attack complexes or enhance phagocytosis. So I don't want you to think of innate immunity and adaptive immunity as two separate, unrelated branches of defense. And in fact, based on this slide, you can see that the two kind of crosstalk, right? The two kind of feedback on each other to enhance the immune response, right? Antibodies are supposed to be the adaptive immunity, but once you form the antibody, it could feed back to the innate immunity and cause you to make more membrane attack complexes, and then it will also enhance phagocytosis. So it's more like a network of response rather than a linear series of events. So those are the three ways that antibodies can help with the infection. Here is another important concept. Antibodies are proteins, and sometimes they are much smaller than the antigens that they recognize. So not all antibodies are created equally. In fact, the antibodies that you create are based on shuffling your genes. Okay, it's kind of like shuffling a deck of cards and then dealing out 6 cards, and you're gonna get different cards every time, right? Sometimes you might get the same set of cards, but there are so many different combinations, right? So the different combinations that you get by shuffling your genes will actually cause the shape of the variable regions to be different, and hence the ability to recognize different targets. And so not all antibodies are going to have the same efficacy when it comes to binding to their targets. So some will be stronger, it will bind to targets with 2 arms, and some will be a little bit weaker. So it turns out the antibodies that are going to be most effective are the ones that binds to the target with higher affinity, right? So the specific location on the antigens that the antibody binds to is what we call an epitope. Okay, so when we create a vaccine, for example, we want to use an epitope that is very immunogenic. Okay, that will create antibodies that have strong binding. So now that we know how antibody works, let's talk about how antibodies are made, and we're going to do that in a class note. So let's say we are infected with some red bacteria. Okay, so these are red bacteria right here, and we are being infected with them. So what happens is your innate immunity is going to kick in and try to get rid of them. So you're going to have your inflammatory response, you're going to have your complements, and you're going to have your phagocytes. So here I'm going to draw a phagocytes, okay? And I'm going to use macrophage as an example. So here is my macrophage. And again, I encourage you to draw this together with me. It's going to help you understand things a little bit better. So we all know that macrophage is an example of phagocytes. So it's going to do phagocytosis, and it's going to basically eat the red bacteria. Okay, so we're going to eat it. It's going to put it in a vesicle. Okay, so that's what we know so far. What I did not tell you previously is that the macrophage can actually do something called antigen processing. Okay, antigen processing. So in antigen processing, what happens is that the macrophage will take part of the bacteria that it has engulfed. It will break it down, and then it will put the bacterial antigen on its surface. Okay, the macrophage is really proud of what it has done. It's very happy that it ate the bacteria, and it wants to show everybody. Okay, so it will show everybody by putting the bacterial antigens on its surface. So these red circles are bacterial antigen. Okay, so this process is called antigen processing. Right? So let's go ahead and skip to another thing for now. Okay, we're gonna come back to this in a second. From your bone marrow, okay, from your bone marrow, this is your bone marrow. It's gonna be in the middle, I guess. So your bone marrow, that's where all your white cells are produced. So from it, we are gonna have something called naive T cells. Okay. So naive T cells are basically T cells that are not exposed to foreign antigens yet. Okay, they're naive. They don't know, they don't know anything. Okay, they don't know the ethos of the world, so to speak. Okay, that's why they're called naive T cells. And you're gonna have different types of naive T cells. Okay, you're gonna have a naive T cells with a blue receptor. Maybe you're gonna have a naive T cells with red receptors, and all these receptors are going to be created by gene shuffling. Okay, so it's all random. You might have one with yellow receptor, you might have one with green receptors, and so you get the idea, right? So each of these naive T cells will have a unique receptor. And again, you get this receptor by shuffling your genes, okay? By splicing different introns in and out and connecting different exons together, and you will be able to get a unique receptor, okay? So now, back to our macrophage. The macrophage has just engulfed a bacteria. It's very happy about it. It puts it onto the surface, and now is going to show these naive T cells, okay? It's going to try to find a naive T cells that has the receptors to recognize the same bacteria. Okay, so this macrophage is going to come and show it to this cell. So are you going to be able to help with the infection? Well, because this naive T cell has the blue receptor and it's not suitable for what we are currently having an infection from, so this one is not going to help. Okay, and then it's gonna show it to the next one, and because it has the same receptor, this will be the chosen one. Okay, so the process of showing the bacterial antigens to these naive T cells, this is called antigen presentation. Antigen presentation. So not every macrophage can do antigen presentation. Macrophage can, okay? Neutrophils cannot, for example. So when a cell is able to do antigen presentation, we call them an APC, antigen presenting cell. So a macrophage is an example of an antigen presenting cell, APC. Okay, so back to here, the macrophage, acting as an APC, it would show the antigens to all these naive T cells, and then after locating the correct naive T cell, it's going to activate the naive T cell. So when you become the chosen one, you are going to undergo clonal expansion. Okay, so clonal expansion basically just means a lot of mitosis. Okay, so this chosen T cell is going to clone itself, and you are going to have a lot of them. So all of these cells will be identical. They all came from cell division mitosis of this one, and they will all have the same receptor. And in this case, we are looking for the red receptor that would help deal with the red bacterial infection. Okay, so this here, they are a clone, a bunch of clones of the chosen naive T cell. So after you cloned yourself, after the naive T cell clone themselves, they could differentiate, they could transform into one of four things. Okay, so now first of all, they could become what we call a killer T cell. Okay, killer T cell, not to be confused with the natural killer cell. Okay, so this is different. This is killer T cell. So killer T cell, which is also known as cytotoxic T cells. Okay, so they are going to transform. So there, I'm going to draw another cell here, and they're killer T cells. I'm gonna give this T cells a little weapon here. Okay, and so the cytotoxic T cells here, their job is to destroy infected cells. Destroy infected cells. Okay, and they could do that by secreting something called porphyrin. Okay, so porphyrin is a molecule that will insert holes in the membranes of the target cells. Okay, so don't confuse this with the MAC that we were talking about in the complement earlier. Okay, these are separate types of chemicals, and they would insert holes into the infected cell. So if a cell is infected with a virus, for example, then the killer T cells is going to identify them and put holes in them. Okay, so that is killer T cell, and it's a one-way transformation. Once the naive T cells becomes killer T cells, it will stay that way until it finishes its job. Another choice that it has is it can become what's called a memory T cell. Okay, so a memory T cell. So the memory T cell, I'm gonna give it a memory bubble. Okay, it remembers. Okay, so the memory T cells will remain in the body, for a very long time. Okay, for a very long time. Theoretically, it can remain there for a lifetime. Okay, but sometimes they do go away, and then you might lose the immunity a little bit. But for the most part, they remain in the body for a very long time, and is going to be able to create a rapid response if you are infected by the same pathogen. Okay, so next time if you get infected by this red pathogen again, then you don't have to go through this whole selecting process. Okay, you recognize it, you can skip directly to differentiating into the killer T cells and to help deal with it. Okay, and then there is another one called the suppressor T cell. Suppressor T cell, and the function of the suppressor T cell is basically to stop the immune response when this is all over. So I'm gonna give this guy a stop sign. Stop. Okay, so this will stop immune response. You don't want to keep on having mitosis when there is nothing to fight, right? Uncontrolled cell division is quite a dangerous thing. It can lead to cancer, right? So stop immune response when the infection is cleared. Okay, and then there is an other type of cells that you can become. The naive T cells can become, and it is called a helper T cell. Helper T cell. Okay, so helper T cell, I'm gonna give it a helping hand. Okay, this is a hand. There we go. And what does helper T cells do? All right, so we're gonna get to that in a second. But let's go back to our bone marrow here for a second. The bone marrow is going to also create a second group of cells called the naive B cells. So similarly to the naive T cells, these are B cells that have not been activated. They have not been exposed to foreign antigens. And similar to naive T cells, each of the B cells created are going to be different. Okay, so we might have a B cells that have the ability to create green antibodies. We're gonna have one that can create orange antibodies, and we're gonna have one that could create blue antibodies maybe, and we're gonna have one that can create red antibodies, right? So of course it's possible that you might not have the right naive cells for the infections that you're experiencing. And in those cases, you're just going to take longer to recover from the illness, right? You're going to have to wait for the body to eventually create that particular naive cells by chance. And so what happens is the helper T cell is going to activate these naive B cells. Okay, it's going to help activate the correct naive B cell. Okay, so it's gonna check with this one. Do you have the right receptor to the right antibodies to deal with the infection? And green is not the one we're looking for, and orange is not the one we're looking for. Blue is not the one we're looking for, and so it will find the right one. And when it does, this would be the chosen one. Okay, and just like before, when you are the chosen cell, then you are going to undergo clonal expansion. Okay, and that basically means you're gonna make a lot of copies of yourself, okay, through mitosis. Okay, so they're supposed to be the same, and they will all have the ability to make antibodies. Okay, antibodies. There we go. Then what happens is, just like before, these clones, they would have different choices. They could differentiate, they could transform into different cells. So over here, they could transform into what's called memory B cells. Memory B cells. And it's the same function as before, right? They are going to stay in the body for a long time, and next time you get infected by the same thing, you can bypass all the steps from before and skip straight to this mass production. And you are going to create something called plasma cell. Okay, plasma cell. Okay, so a plasma cell is basically antibody factory. Okay, so here is the plasma cell, and it's going to mass produce antibodies. Right, so you have all these antibodies coming up. And these antibodies are going to be tailored to the infection that we have, and in this case, it would be tailored to attack these red bacteria. Okay, so how would the antibody help? Well, it's the things that we talked about, right? They could neutralize the toxin if the bacteria is producing toxins. They could form these immune complexes, right, and cause things to clump together, right? They could encourage the formation of membrane attack complexes, which would then cause holes to form on the bacteria, right? So by creating these antibodies, we are going to be able to fight off the infection. So this whole process takes about like a week or two, right? Sometimes that's how long it takes before you feel a little bit better. So a couple other things that I want to add to the diagram. So these cells, the cells that the naive cells can become, the killer T cells, memory T cells, suppressor T cell, and helper T cells, they are called effector cells. So there are 4 different types of effectors for naive T cells, and there are 2 types of effectors for naive B cells. So let me just quickly summarize it for you down here. We have the naive T cell. And in order to activate the naive T cell, we would need to have an APC, okay, an antigen presenting cell. Okay, so in this case, we talked about macrophage. There are other APCs in the body, but we're just going to keep it simple. So once you have been activated, once you have been chosen by an APC, then they could become, the killer T cell. They could become the helper T cell. They could become the memory T cell, or they could become a suppressor T cell. Okay, all these cells are called effectors. Okay, so if I ask you what are the effectors of naive T cells, they are it. Okay, so the immunity that you have, the immune response that you have from the T cells, we call this cell-mediated immunity. Okay. On the other hand, we have naive B cell, okay? And to activate the naive B cell, to choose the correct naive B cell, you will need to have a helper T cell. Yeah. So once the naive B cells are chosen by the helper T cell, it will differentiate into memory B cell, and plasma cell. Okay, plasma cell. So these are antibody factories, right? So again, these are called effector cells. These are the effectors for naive B cells. Okay, and immunity that you have from B cells, we call this antibody mediated immunity. Okay, or sometimes in textbooks they call this humoral immunity. Okay, so it's just two branches of adaptive immunity: cell-mediated and antibody-mediated. So this could happen anytime you encounter a foreign antigen. Okay, you might remember Rh sensitization, right? So suppose this is not bacteria. If this is just like a red cell with Rh on it, then you would go through the same process, and in the end, you would the end result would be you now have anti-Rh antibodies, right? And so you are sensitized. You created memory cells, memory B cells and memory T cells against the Rh factor. So that's why the second time you get transfused with positive blood, you are going to react against it, right? You already have these antibodies. You're going to have a cross reaction. Okay, so that's basically how antibodies are produced, and it's quite a long process, but hopefully you understand it from the video.